Olfaction. Olfaction represents an ancient evolutionary critical physiologic system. In humans, the chemosensation mediates safety, nutrition, sensation of pleasure, and general well-being. Factors that affect human olfaction included structural aspects of nasal cavity that can modulate airflow and therefore odorant access to the olfactory clips. After signals are generated, olfactory information is processed and coded in the olfactory bulb and disseminated to several areas in brain. Here we are going to discuss about the functional anatomy of nasal cavity for olfaction pathway and the areas of brain responsible for olfaction. Olfaction is the most important of the sense in many mammalian species as it provides information about food that is essential for individual survival and reproductive success. A major route for bringing molecules to the olfactory epithelium, especially from the food in the mouth, is called the retronasal pathway. More than that, the volatile substances from food diffuses and pumped during mouth movements or pass during exhalation from nasopharynx into the rear of the nasal passage and thence it to the vicinity of the olfactory epithelium high in the nose. Both sexes use olfaction to transduce chemosignals of relevance for mate recognition and reproductive behavior. In fact, the animals display great interest in the body odors. Now, commercially, we know there are different types of perfumes we use for women and men. So, in this chapter, we focus on delineating the relative involvement and the interaction between two primary structures that exist for perceiving such socially relevant chemosignals, that is called pheromones or semiochemicals, and the main accessory olfactory systems which receives input, that is the main olfactory epithelium and the vomeronasal organ for mammals. A very true fact, all we know, and it is scientifically also proved that memory is triggered by the scent, which has a strong emotional connection and appears more intense than other memory triggers. We are going to discuss about the structure of olfactory epithelium. This is a schematic diagram of nasal cavity where we can see the olfactory mucosa or olfactory epithelium is situated. Now this is a nasal cavity and whole thing is nasal cavity. This and in this nasal cavity, the olfactory epithelium or mucosa, whatever, it is situated at the dorsal posterior recess of nasal cavity. This is the olfactory epithelium or mucosa. And in this mucosa, the olfactory chemoreceptor, a specialized part of the nervous system, these are situated. And these olfactory chemoreceptors are bipolar cells. Check it here. Now, this is olfactory epithelium or the mucosa, the whole blue thing. Okay, and in this mucosa, the olfactory chemoreceptor, which are actually bipolar cell. These are bipolar cells. And the dendrites of this bipolar cells has a knob-like structure. And this knob-like structure are containing some cilia. These cilia are unmyelinated process which are 2 micrometer in length and 0.1 micrometer in diameter, which contain specific receptor for odorant or odorant receptor. Now, what is happening to the axon? The axons of this olfactory neuron pass through the cribriform plate. This is the cribriform plate of ethmoid bone which is situated at the base of the skull. And these axon, after passing through this cribriform plate, synapses in the olfactory bulb with the second order neuron. And this olfactory bulb, this is the olfactory bulb, and this olfactory bulb is a portion of a cerebral hemisphere of the brain which is located at the base of the cranial cavity just below the frontal lobe. Now this is the olfactory bulb. Here you can see the neurons of the first order bipolar cells are getting synapsed with the second order neuron, and the second order neurons are going towards the cerebral cortex and a different part. This is a simplified form of that diagram only. Here you can see the different parts in zoom. What is happening? This is the bipolar cell which is present in olfactory mucosa or olfactory epithelium, supported by supporting cell or the sustentacular cell. These are the bipolar cells, and the cilia of these bipolar cells are jutting outside to the external wall, where the odorant are binding with the odorant receptor which are present in the cilia, and this way the signals are passing through this nerves, the first order neuron, and crossing the cribriform plate of ethmoid bone. This is the cribriform plate of the ethmoid bone, and then the first order neuron are ending and getting synapsed with the second order neuron inside the olfactory bulb. And the olfactory bulb, from there the second order neuron are carrying the sensation towards cerebral cortex. Now we are going to discuss about the basic neuronal circuit in olfactory bulb. Uh, this is a schematic diagram where there are different type of cells or the neurons are present. First, we'll know about the CP. What is CP is cribriform plate. Then here we can see there is written OG. OG stands for olfactory glomerulus. What is this? Here, the first order neuron are synapsing with the second order neuron. Okay. Next, there are two type of neurons present in the olfactory bulb. One is mitral cells. Here is a mitral cells, and next is a tuft cell. This is a tuft cell. This again, these are bipolar cells. This mitral cells and tuft cell are synaptic with the first order neuron. Okay. In addition to mitral and the tuft cells, in the olfactory bulb contains two other kind of cells. What is PG? PG stands for periglomerular cells. This is periglomerular cells. Check it here. It is connecting two glomeruli, means this and another glomeruli or the another neuron. This are inhibitory neuron connecting one glomerulus to another. Clear? And then another type of cell or the neuron, it is called granule cell, which have no axons and make reciprocal synapse with the lateral dendrite of both mitral and tuft cell. At the synapse, the mitral and the tuft cell excite the granule cell by releasing glutamate. Okay, this mitral cell and the tuft cell are secreting glutamate and exciting granule cell. Granule cell in turn inhibits the mitral and tuft cell by releasing GABA. This is the level where granule cell secretes GABA. Now, each glomerulus, this is a glomerulus, this olfactory glomerulus includes the apical dendrites of approx 25 mitral cells, which receive innervation from 25,000 olfactory receptors. So what is happening? The neurons, the first order neurons are converging to the mitral and the tuft cell. So this degree of convergence presumably serves to increase the sensitivity of mitral cell to ensure the odor detection, because there are so many odor and that should be perceived with a particular type. So this convergence helps this odor detection. Because of each glomerulus is specialized by being the target of the specific olfactory afferent for a unique combination of the odor qualities, this appears to be the way of enhancing stimulus contrast. Means it is contrasting. It's some other a fragrance or it's a good smell or a bad smell. This is very important. It is happening around this olfactory glomerulus level. In addition, it provides a mechanism for adaptation to continuous stimulation. Now, what are the parts of the brain are involved in smell detection? After the olfactory receptor cells, this olfactory nerves enters to the olfactory bulb. From the olfactory bulb, the neurons enters as a olfactory tract. This olfactory tract divides into two. One is medial olfactory stria and another one is lateral olfactory stria. The medial olfactory stria or area consisted of several nuclei located at the mid-basal portion of the brain immediately anterior to hypothalamus. And most conspicuous of this nucleus is septal nuclei, which are the midline nuclei that feed into hypothalamus and are the primitive portion of brain's limbic system. The mainly from the septal nucleus, this is the neurons ends into this hypothalamus part and mainly the primitive, very primitive parts, and that's why this tract is called very old olfactory system. Now coming to less old or the lateral olfactory stria. This lateral olfactory stria, there is a two types by time of discovery. This is one is less old olfactory system and another one is newer system. As we all know, this is the smell sensation is only a sensation of the sensory pathway which doesn't go through or pass through a relay nucleus thalamus. But the newer system, it detected that the some fibers passes through thalamus too. Now, before that, coming to the less old olfactory system. This lateral olfactory area is composed of mainly the pre-piriform cortex. The piriform cortex there are piriform cortex, entorhinal area, and amygdala. Actually, from this piriform cortex, the signal passed into almost all portion of the limbic system, especially into less primitive portions such as the hippocampus, which seems to be the most important of the learning to like or dislike certain foods depending on one expectation or the experience. And the other part, that is a newer system, it passes through thalamus, passing to dorsomedial thalamic nucleus, then to lateral-posterior quadrant or orbitofrontal cortex. As we all know, that smell perception is very much related with emotions and memory. and sexual preferences also. So the most of the signals from the olfactory bulb, which is going to hypothalamus nuclei and the nucleus of limbic system. Now this is simplified structured, or the simplified diagram, whatever we have discussed in the debate regarding this olfactory pathway. Here we can know about where are the information going from the mitral cells and also the tuft cells. So here we can see there's a diagram of olfactory bulb, and there are two types of cells. One is mitral cell and another one is tuft cell. From the mitral cells, mainly the signals are projecting to the five regions of the olfactory cortex. Where they are going? They are going to the olfactory nucleus. First is the olfactory nucleus, then it is going to the olfactory tubercle, then to piriform cortex, then it is going to the amygdala and entorhinal cortex. Okay, and from the olfactory nucleus, it is again going towards a contralateral olfactory bulb. That is very important pathway. This connecting two olfactory bulbs are connected by each other from the anterior olfactory nucleus to the contralateral olfactory bulb. Now, from the tuft cell, it is projecting mainly towards the anterior olfactory nucleus and then olfactory tubercle. And these are the main sources from where they are coming: mouth, mitral, and the tuft cell. The mitral cells is also present in olfactory bulb. So accessory olfactory bulb, which we are going to discuss in the later slides. The mitral cells, which are present in the accessory olfactory bulb, project only to amygdala, and conscious discrimination of the odor depends only mainly to the neocortex. That is, which are the parts of the neocortex which are mainly involved in conscious discrimination. Those are orbitofrontal and the frontal cortices. The emotive aspects of the olfaction derives from the limbic projection of amygdala and towards the hypothalamus. So mainly it is these two nucleuses are mainly involved in the emotional part of the smell sensation, and ultimately it is also related to memory. Now we'll discuss about how the odor is discriminated, and what is odorant binding protein. Actually, it is very surprising that uh, there are 10,000 different types of odor present. But the psychological studies, it has been seen that there are a few types of odorant which are very common, which is expressed by the studies. One are the chemoferous and musky, floral, pepperminty, ethereal, pungent, and putrid. Though these are the most common, but there is 10,000 different of odors. Now it is very surprising, and as we know, for example, there is methyl mercaptan, which is present in the garlic. Even if it is present in the less than 500 picogram per liter in the air, it can be sensed by the sense organ. How it is possible? As we know, this sense organ, such as the olfactory epithelium, at its brain representation, which apparently lacks a high degree of complexity, but still it differentiates 10,000 types of different odors. How it is possible? And it is possible first by the gene that code for about 1,000 different types of odor, and receptors make up the largest gene family so far described in the mammal. Okay. And another important thing which is responsible for the discrimination of the different type of sense is there is a neuronal organization of the olfactory pathway. Although there is millions of the olfactory sensory neurons, each expresses only one type of thousand different odorant receptors. And each neuron projects to one or two glomeruli. This actually different projection to the glomeruli. This provides a distinct two-dimensional map in the olfactory bulb. And another one already we have discussed, that is a olfactory glomeruli demonstrate the lateral inhibition mediated by the periglomerular and the granule cells. These are very important for the discrimination of the different type of the smells by the different person. Now, another thing is very important. This odor producing molecules are generally very small, containing 3 to 20 carbon atoms. Molecules with the same number of the carbon atoms, but the different structural configuration with for the different odors. Now, how it is coming nearby to this receptor? There is one called odorant binding proteins. These proteins concentrate the odorant and transfer to the receptor. So it is very easy to bind this odorant to the odorant receptor. In this slide, we will get to know what is a signal transduction process happening in olfactory cilia when the odorant receptor comes near olfactory cilia. This odorant receptors or the odorant substance are coming in contact with the olfactory membrane. First, it diffuses into the mucus that covers the cilia. Then, in the cilia, there is a special type of receptor is present. And this receptor is nothing but the 7 transmembrane protein, or it is a G protein coupled receptor. This is the G protein coupled receptor. This is the 7 transmembrane protein, which is still not coupled with G protein. Actually, this is the inactive G protein. Clear? And another thing in the cell membrane of cilia is present is the transmembrane enzyme or the protein called adenylyl cyclase. Another one it is present is cyclic AMP dependent sodium calcium symport, which is present in the ciliary membrane also. Now, whenever the odorant binding with the 7 transmembrane protein, which is present in the ciliary membrane, there is conformational changes and it is activating this G protein. After activating, what is happening? The alpha subunit of this G protein is getting detached. This detached alpha subunit then stimulating the adenylyl cyclase enzyme, which is present in the ciliary membrane. After adenylyl cyclase is getting activated, this is converting or hydrolyzing the ATP to cyclic AMP. And then cyclic AMP dependent sodium and calcium channels or the symports are getting opened. Okay, what is happening here? Here we can see this is a cilia which is present, and there is opening already. It is open state because of the alpha subunit is stimulating the cyclic AMP, and because of the resting membrane potential of the cilia is minus 50 millivolt. As soon as the sodium and calcium is entering, there is positivity inside. And another reason there is an increase in positivity is this: there is an opening of chloride channel too. Because of the chloride, it effluxed from the cilia inside the cilia towards outside, so more positivity creating inside. And because of this resting membrane potential is moving towards a positivity, there is a depolarization. And because of the depolarization, there is in the cilia, there is a production of graded potential first, and this graded potential are getting summated. Then after summation, it is producing an action potential, which is action potential can move unidirectional movement, and it is moving towards the olfactory bulb, and the spike potential through this neuron, it is moving towards olfactory bulb. In rodents and the various other mammals, the nasal cavity contains another patch of olfactory epithelium located along the nasal septum, and it is called vomeronasal organ or the Jacobson's organ. The Jacobson organ is organ for chemoception. This structure is mainly concerned with the perception of the odors that acts as a pheromone. And this vomeronasal organ, sensory organ, project to the accessory olfactory bulb, and from there the primary area to amygdala and hypothalamus. And what happened to this uh, organ to human? Is it present? That is a very important question. The major function of this vomeronasal organ, it has been seen that this example in the pregnancy of mice. Pheromones of the male from a different strain prevent the pregnancy as a result of mating with that male. And in case of human, it is not very developed. Here we can see there is a patch kind of epithelium is present, which is mainly at the anterior-inferior third of the nasal septums. And this organ contains specialized olfactory sensory cell. And some scientists say it is bipolar cells are also present. Actually, it is an accessory organ for human, and it may detect the pheromone. And as we have discussed, it is mainly developed in the rodents and mammals, but except in human. What happened in human, the vomeronasal organ develops in utero, but subsequently it regresses. And all studies agree that it is non-functional. Instead of that, the vomeronasal organ, the human, there is a accessory olfactory organ is present. The diagram is saying that, and the afferent fiber mainly reaching to the amygdala and hypothalamus, has a close relation with the smell and the sexual functions, and which we see in the perfume industry. And the advertisement usually comes, it's very much in the internet or the posters. There is a different kind of perfumes for the males and the female, which will be attracted to other sex if you use that perfume. Is it really is because of this non-functional vomeronasal organ or the accessory uh, olfactory organ? Still the research is going on. And two studies prove that the adult accessory olfactory organ have reported the presence of the bipolar cells. Now, what is the relationship between the olfaction and memory? Or otherwise, we can say it is the olfactory memory. The olfaction is generally assumed to play a role in the humans that have a unique status for eliciting memory. And sometimes it happens you are sniffing or getting a smell of a flower, you are remembering something, or a food which is exactly smells like your mom's food. So it brings about a memory. What is the relationship between that and what is the connection? That is a very interesting part. The Proust phenomenon. So some scientists says it is called the Proust phenomenon. The odor memory in the human resides in the imaging power of odors to vividly triggers the evocation of autobiographical experience. In animals, it plays some very important and critical role because of the environmental orders plays a vital role. Where is the for the mice? Where is that food? And it has been also scientifically proved that mice will go in the same pathway which has been formed the memory that where is the food is, and also it is responsible for their survival. So they may won't go to that particular area where their danger is. So olfactory bulbs output neurons have a connection with the amygdala and their hippocampus. The connection will show in the next slide. The main olfactory bulb makes a dense monosynaptic contact with nuclei of the corticomedial amygdaloid group, including the periamygdaloid cortex. Continuation of the last slide. There is an anatomical and electrophysiological studies have shown that there is an olfactory bulb and the piriform cortex, saying their direct connection to the entorhinal cortex, which is responsible for mainly the memory production or the consolidation. There is a diagram. You can see the olfactory bulb. From the olfactory bulb, it is going to the piriform cortex and lateral entorhinal cortex, ultimately which is going to the amygdala and the hippocampus. So these are the loops for consolidation of the memory. So there is a strong uh, connection between the olfaction and memory. Last, we are going to the clinical aspect or the applied aspect of olfaction, the disorder of olfaction. Dysosmia, cacosmia, or parosmia is a disorder or the perverted smell perception to odor stimulation. Phantosmia, or the olfactory hallucination, is a dysosmic sensation perceived in the absence of any odor stimulation. Olfactory agnosia, an ability to recognize an odor sensation even though the olfactory processing, language, and general intellectual functions are perfectly intact. It happens in the stroke. Uh, Presbyosmia, decline the smell sense with the edge, and osmophobia, dislike or the fear of some particular smell. Complete loss of olfactory functions. There are two types. One is like a hearing. There is two types of complete loss of olfactory function. One is a conductive. The causes for the conductive impairment of the olfaction is a chronic nasal inflammation or the multiple polyps in the nasal cavity. And another one is sensory neural. This is due to the olfactory pathway, the olfactory neuroepithelium, and the connection. May the reason behind this are the airborne toxins or the tumors or the seasons. So here we end the physiological aspect and the molecular basis of olfaction.